Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the microscope. Today, we are starting our lesson on cell biology. We will begin by studying the different types of microscopes. We will look at the light and electron microscopes and determine the functions of both. By the end of this lesson, you should be able to describe the uses of each of the different parts on the light and electron microscope. Let us start by discussing how microscopes are used to study cells. The microscope is one of the most important tools scientists use to study living organisms. Cells are the building blocks of life, and most can only be seen under a microscope. Microscopes are instruments for magnifying specimens. They have been used for over 300 years to look at and study cells. As microscopes have improved, so has the understanding of cells and therefore of living organisms as well. There are two main types of microscopes that are commonly used: the light microscope and the electron microscope. We will be learning about both, but let us begin by discussing the light microscope. The light microscope uses a beam of light to form an image of the specimen. Slide cells, tissues, or individual organisms are often the specimens that are viewed using a light microscope. These very small particles of biological matter are stained or treated to help the observer see the specimen more clearly. Stains are chemicals added to the slide tissues to make the cells, or parts of the cell, easier to see. The cells that have been stained or treated are dead. The cells are fixed or treated with chemicals so they do not decompose or decay. A live specimen can be viewed under a light microscope as well. However, they are not as easily observed as the treated specimens. Now, let us discuss how the microscope works. A specimen is placed on an area called the stage. The specimen is lit from underneath. Some light microscopes have a single lens. Other microscopes, called compound microscopes, have two lenses, an eyepiece lens and the objective lens. A compound microscope produces a much more magnified image than a single lens. In a compound microscope, the light passes through the specimen and through the objective lens. The image is produced at the eyepiece lens. It is highly magnified, upside down, and is viewed right to left. The magnification of the specimen is calculated by multiplying the magnification of the objective lens by the magnification of the eyepiece lens. So if, for example, the magnification of the objective lens is 50 times and the eyepiece lens is 10 times, then the overall magnification will be 500 times. We have covered quite a bit of information already, students. Let us do an activity to discuss what we have learned thus far. Students, please determine whether the following statements are true or false.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. How did you do? Let us go over the answers together. The light microscope does use a beam of light to form an image of the specimen. Stains do make it easier to observe a specimen. Stains do not encourage the specimen to decay or decompose. By applying stains to a specimen, it makes it easier for the observer to view the specimen, and it prevents it from decaying. A single lens microscope is not more powerful than a compound microscope. A compound microscope is more powerful than a single lens microscope. The magnification of a specimen is not calculated by adding the eyepiece lens magnification and the objective lens magnification. It is determined by multiplying the magnification of the eyepiece lens by the magnification of the objective lens. When using a light microscope, information about the specimen is obtained by using the light in different ways. Dark field illumination is a method where the background is dark and only the specimen is illuminated. This is useful for showing smaller structures within the specimen. Now, let us look at the electron microscope. As we have learned, light microscopes use light to form an image. An electron microscope uses beams of electrons to form an image. Light has a limited resolving power. Resolving power means the amount of detail the microscope is able to show. Electron microscopes are able to show much more detail. They give a much higher magnification and resolution. The electrons act as light waves, but with a smaller wavelength. As the wavelength gets smaller, the resolving power is increased. Therefore, the electron microscope can resolve the detail of a specimen down to 0.3 nanometers. Samples that are viewed under the electron microscope must be fixed, stained, and thinly sliced in a similar way to the samples viewed under a light microscope. However, the materials and stains used are very different. An electron microscope works by having electron beams focused by magnetic lenses. A series of magnifications forms the image. The image is formed as electrons are scattered by the biological material of the specimen. Unlike a light microscope, you do not look directly into an electron microscope. Electronics attached to the microscope produce the image on a television screen. The image can be saved as a photograph called an electron micrograph. Electron micrographs show the surface of the cells. They also show the details of the structure, allow for a greater depth of focus, and provide a three-dimensional view of the specimen. Students, before we conclude our lesson for today, let us do an activity to review what we have learned. Please write down 3 characteristics of the electron microscope that differentiate it from the light microscope.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, everyone. I am sure you came up with some interesting responses. Some characteristics you may have written down could be that the electron microscope uses electron beams to form an image, while light microscopes use beams of light to form an image. You may have also said that the electron beams are focused by magnetic lenses. You may have also said that electron microscopes produce electron micrographs that are viewed like a photograph, while the image from a light microscope is viewed through the eyepiece. These are only a few of the correct answers. You may have written down different answers that could be considered correct as well. This brings us to the end of our lesson. Today, we learned about light and electron microscopes. We learned how they are used to view specimens. Until next time, thank you, teacher. Thank you, students.